Okay, thank you, Eddie, and the organizers of this wonderful symposium for inviting me to participate and present our data. It's really remarkable to be here in this incredible location. I'm going to share with you the data that we got in our lab over the last few years on responses to hypoxia. Okay. Hypoxia is a trigger for a wide array of physiological and pathological conditions, including angiogenesis, erythropoiesis, vasodilation, apoptosis, cell proliferation and survival, and metabolic adaptations. These responses rely mostly on, and we conduct our studies in the fruit fly Drosophila melanogaster mostly. These adaptations rely mostly on a single transcription factor that is called HIF, from hypoxia inducible factor. That is, it's a heterodimer of alpha and a beta subunit that is widely conserved in evolution, in which the Drosophila homologs are a protein called Sima and Tango for the beta subunit. They recognize HIF responsive elements on the enhancers of these of the target genes, and they activate a few hundred of downstream genes that mediate the adaptive responses. The regulation is exerted at the level of the alpha subunit, while the beta subunit is constitutive, and the alpha subunit is regulated by oxygen, and the regulation depends essentially on an enzyme, a prolyl-4 hydroxylase, that utilizes molecular oxygen as the substrate for the catalytic reaction, so that hydroxylation of a key prolyl residue on this alpha subunit is catalyzed when oxygen is available. And this hydroxylation enables the recognition of a protein called the VHL, which stands for the von Hippel-Lindau tumor suppressor factor, which in turn is the substrate recognition subunit of a multimeric E3 ligase complex that in turn promotes uridination of the alpha subunit, which ends up in rapid degradation at the 26S proteasome. When oxygen is scarce in hypoxia, this degradation does not occur. The protein becomes stable. It forms an heterodimer with the beta subunit and can promote the expression of the hundreds of HIF target genes. A few years ago, we decided to try to define new regulators of this key transcription factor of HIF, and to do so, we utilize Drosophila's cell line called S2 line, for which we designed a reporter element in which the firefly luciferase is controlled by an enhancer derived from a murine gene, the lactate dehydrogenase gene, that is inducible by HIF. We co-transfected with cells with a constitutive reporter, a RNA-Laz luciferase, in which that is controlled by an actin promoter. And as you can see here in this graph, as oxygen concentration goes down, luciferase activity goes up, so the reporter works. And interestingly enough, it's not only regulated by oxygen, but also by iron, so that if we include in the cell culture this iron chelator, the ferrooxyamine, the reporter is also induced. And the reason for this is that the prolyl hydroxylase that exerts its function as an oxygen sensor is an iron-dependent protein, so that if we chelate the iron, it responds as if there was hypoxia. So this is very important. What we did next is to treat the cells with RNAi against the two subunits, the alpha and the beta subunit, which are Sima and Tango. And as you can see here, the strong induction that occurs in hypoxia is completely blocked. The same applies to DFO, to the iron chelator. So we were in a position to do this, the screen with RNAi against all the 22,000 genes of the Drosophila genome. And we did that in collaboration with the Drosophila RNA Screening Center that is located in Harvard Medical School. So the screen went well. After 3 phases of election, we ended up with 21 genes that were not previously associated with hypoxia biology, along with other genes that, of course, were known to regulate HIF, including the HIF subunits themselves. And among these hits, we got a little bit of everything, including genes controlling protein translation, chromatin remodeling, microRNA biology, transcription, RNA processing, and miscellanea. The rest of the talk will be focused on the microRNA machinery. I will not show the data now, but we have been able to show that not only Argonaut1 was a hit of the gene. Argonaut1 is the homologue of Argonaut2 in mammals. We have been able to prove that also DROSHA, DICER, and GW182, classical genes that are required for operation of the microRNA machinery, were all required for the hypoxic response. So what we did next was we hypothesized that if the microRNA machinery was required for the hypoxic response, probably one or more microRNAs were required for this response. And since microRNAs are known to be inhibitory of the target genes, we postulated that this was indirect, and there was an X protein blocked by this microRNA that in turn, the X protein inhibited Sima, provoking the increase of the hypoxic response. So that the question was, what was the identity of these one or more microRNAs? And the experimental approach was the following. If by blocking the microRNAs, the X protein was supposed to go up and Sima was supposed to be inhibited and the hypoxic response was abrogated, then perhaps by increasing the amounts of this microRNA, the X protein would go down, Sima would go up, and the hypoxic response would be enhanced. So what we did is an overexpression screen of microRNAs of the encoded in the Drosophila genome, And we did that in vivo, in transgenic lines. We collaborated with the lab of Norbert Perimon in Harvard, who had created a whole collection of transgenic lines overexpressing these microRNAs. And we created a transgenic line expressing beta-galactosidase reporter, very similar to the one that I described in the cell culture, controlled by the same enhancer, the LDH gene from mouse. So we overexpressed all these microRNAs at very mild hypoxic conditions to help the system to be induced, and we let the flies egg lay in these sort of devices. We utilized negative controls, a positive control that was the RNAi against the prolyl hydroxylase that we call fatiga in the flies. We got many, of course, negative results, and luckily enough, we got some positive results that were the following. We got miR-190, miR-272, and two other miRs. The rest of the talk will be focused on the activity of miR-190 and how it regulates the response to hypoxia. Here you can see the quantification. All these microRNAs indeed induce the expression of the hypoxia-inducible reported at very high frequency. miR-190 is encoded in an intron of a genomic locus called RHEA. RHEA is a cytoskeletal protein. This kind of microRNAs are called miRtrons because they are encoded in introns. And as you can see here, the overexpression of miR-190 is dependent on Sima. Here I underlined in green 190 and in red the genes that block this induction. Here I'm not sure if you can see the small letter. The induction is blocked by the RNAi of Sima, implying that this induction indeed depends on Sima, and as expected, is also blocked by overexpression of the prolyl hydroxylase fatiga. So this implies that indeed miR-190 was working through the fatiga and Sima pathway. We next did the opposite experiment, that was to test what happens in a miR-190 mutant. As you can see here, hypoxia-dependent induction of the reporter is reduced in the mutant. And this is not only true for the reporter, but also for HIF endogenous genes in Drosophila. These are two genes. This is the Hitchcock factor and another gene called HPHB. That, as you can see here, when we overexpress 190, it's upregulated in both cases. This is true. So we did the converse experiment that was to test in these HIF endogenous genes how the hypoxia-dependent induction that you can see here in the white column behaves in the mutant, is clearly reduced. This is true for the two genes that we tested. So we were confident that miR was really controlling the response. The next obvious question was, what was this X protein, the miR direct target messenger RNA. So we looked at several different databases that predict the RNA target genes. And we noticed that in one of them, in the Miranda databases, fatiga, the prolyl hydroxylase, appeared as one of the possible targets. And there were two possible binding sites on the 3' UTR of this gene. So we went on and asked if fatiga was probably, was maybe the direct gene controlled by 190. And since antibodies are not available against the prolyl hydroxylase, we utilize an alternative system. It's reported in transgenic flies, in which the oxygen-dependent degradation domain of HEAF, the one that has the prolyl residue, is fused to GFP under control of a constitutive promoter, so that when this prolyl residue is hydroxylated by the action of the prolyl hydroxylase of fatiga in the presence of oxygen, it's rapidly ubiquitinated and degraded, and if fatiga is reduced, then we will not have this rapid degradation. So the important thing to remember here is that more green signal means less fatiga, and the opposite. Less green signal means more fatiga. While the red signal is constitutive because it's encoded, it's regulated by the same promoter independently. So we did the study in imaginal discs. Imaginal discs are the primordia of the organs in the fly. We did so in the imaginal disc of the wing. And in Drosophila, we have the possibility to express genes in given regions of an imaginal disc, and we did so. We expressed 190 only in the posterior compartment of the imaginal disc, that is the one that will give rise to the posterior part of the adult wing. Please remember, the reporter is expressed all over. So the experiment went well. As we said, more green signal means less fatiga. This is the compartment in which we express 190, and as you can see, we have more green, less fatiga. So overexpression of the microRNA leads to reduction of the level of the prolyl hydroxylase, while the other is just a control. So it was apparently true that that fatiga was regulated by miR-190. So the next important question was to assess as whether this regulating was direct or indirect. So we did this experiment in culture, in cell culture. We fused the luciferase open reading frame to the fatiga 3' UTR, and we did that in two versions, the wild type sequence or a mutated version of the fatiga 3' UTR. As you can see here, overexpression of miR-190 clearly reduces the activity of the luciferase, while when we do the same in the mutated form of the 3' UTR, this does not take place, indicating that miR-190 is really controlling the amount of fatiga, probably at the level of translation. The last question was as whether miR-190 is itself regulated by oxygen. So as you can see here, it is because in hypoxia we have increased levels of miR190. This is also true not only for the mature microRNA, but also for the pre-microRNA. We observed the same for the RIA gene, the one that hosts the microRNA in one of its intron. And all these inductions dependent on hypoxia are independent on Sima. So it's induced by hypoxia in a Sima independent manner. So this is the model. In low oxygen levels, we have inhibition of the oxygen sensor due to the lack of the substrate for the catalytic reaction. This, in turn, promotes the expression of the microRNA that contributes to inhibit the oxygen sensor, strengthening the response to hypoxia. That, in turn, depends on Sima. So that's the scheme. So I want to finish by acknowledge the people who did the work. It was mostly done by Mariana Melani and Ana Laura de Lela Escurra with the help of Maxi Katz. We have been collaborating for many years with Peter Radcliffe from Oxford. And that's it. I'll be happy to take questions.